Good morning, everyone, and welcome to another installment of the MPG Primer series. Today, it's my pleasure to introduce Yunfeng Ruan. She is a staff scientist in Pradeep Nagarajan's lab within the Medical and Population Genetics program at the Broad. She's an expert in polygenic risk scores, or PRSs, and their applications to diverse populations. Her research on PRS began during her PhD at King's College London, where she developed PRS methodologies under the mentorship of Professor Paul O'Reilly. Building on her PhD work, she continued this research during postgraduate training at the Broad, collaborating with Halong Huang and Tiange, thank you, to develop PRS-CSX, an innovative multi-population PRS method. Currently, her research focuses on applying PRS to diverse and admixed populations and leveraging these PRS methods to study heart disease. She has said that questions are welcome throughout the talk, so please come up to the mic so everyone in the room and online can hear you. And the title of our talk today is PRS Methods, Development, and Their Application. Thank you.
Thank you for the nice introduction, and thank you for everyone to come here so early in such a cold weather. So I'm going to talk about the PRS methods, and it's kind of hard to choose a specific topic because there are already so many different methods to calculate the PRS, and it's hard to choose the best one because, as I know in the real application, like each method may have their own like a more favorable situation. So I'm just going through the all the main idea about how, as a method development, about what is important about that method, and I also put the information of the paper. So if you are particularly interested in any specific method, you can look up the paper, or like you can email me, we can discuss. But in this talk in general, I'm just give you a structure to help you to see how we get all these methods. So the PRS, is like, I think it's like almost nearly to say we use it as a proxy of common SNP genetic burden, and it's the math part is very simple. It's a weighted sum of risk allele with the weight being the risk allele effect size. So today I'm going to divide this talk into 4 parts: inventing, calculating, improving, and standardizing and applying PRS. So inventing PRS. Actually, this is a story like maybe started a little bit earlier than you might thought. So back in 2009, when we still kind of like in the darkness about what all the psychiatric disease is. Are they like a heritable, or they purely from environmental or education, this type of stuff? So back then, they gathered the largest possible sample, which is nowadays maybe kind of like a weird, like 3,000 cases and 3,000 control. And of course, we have no genome-wide significant hit like anywhere. And the most significant hit is have this p-value of 10 minus 6. So back then, people are kind of like get nervous and confusing. Should we put more energy and funding to continue investigate into the genetic aspect of the psychiatric disease? So then some very creative researcher come up with our hero, like a PRS. So they inventing PRS as a proof of polygenic genetic basis. So even though none of the SNP hit the genome-wide significant threshold, but the p-value they calculated, like schizophrenia PRS predicting schizophrenia have a very, very significant p-value, as you see, like 10 minus 28. And besides that, they also have some like positive versus negative controls. They use the schizophrenia PRS to predict a highly correlated trait, which is a bipolar, and they also see a significant correlation. But when they predict the non-psychiatric disease, like heart disease, Crohn's disease, and type 2 diabetes, you can see like none of this like association is significant. So basically, they show that the PRS can be used as a genetic, like a proxy of genetic basis, and is quite specific to the disease or to the type of disease. So here is a summary of the first part. So PRS can serve as a proxy of genetic burden even when the SNP statistics are underpowered. So let's go to the next part, the calculating the PRS. How are we calculating the PRS score? So I'll just like first like still go through all these like terms, and here I will use the the term discovery data to represent the summary statistic GWAS that provides the marginal SNP effect size. And then like in the PRS method, we adjust the LD structure and get the adjusted effect size, and we use a data called fine-tuning data to choose the most predictive PRS model. And then like in the testing data, we do all this downstream analysis, like predicting the trait with the PRS or some other analysis. And here are all this, like it's actually just a small part of the existing methods that we can use to calculating the PRS. And there are some like type of it. I also list like for each type, I'll just like list 1 or 2 example. So the most naive one is the clamping and the p-value thresholding. Sometimes we call it CT or PT, like they use clamping and pruning here interchangeably. So PT or CT method. And besides that, we have Bayesian methods, like we adjust the effect size using some like Bayesian method, and also like a penalized regression, and also there's a like a mixed linear regression plus Bayesian method that's based on the individual level data, like both HMM. So here I only like mainly focusing on the CT method, and I'll briefly talking about the Bayesian methods. So the CT method or clamping and p-value thresholding is the most naive and still commonly used method. So here is like the pipeline of how you can calculate the PRS. So I just show like an oversimplified example of like the GWAS data, a Manhattan plot. We can see that like in this part of the genome, we have 3 LD blocks. And from each LD blocks, we choose the most significant SNP or the lead SNP, which is the clamping. And then we can test several different p-values threshold to decide which SNP to keep. And so as we can see, as the p-value threshold be more relaxed, we will include more SNP into the PRS method. So basically, the math of the PRS is kind of like can be written like this. If you have the lead SNP and that SNP have the p-value that pass the threshold, and then you just keep the marginal effect size as is. Otherwise, every marginal effect size just shrink to the 0. So basically, the idea of this method is like we use the least PRS to represent the caudal, the least SNP to represent the caudal SNPs. So here is a kind of like a summary of this method. So it's a hard thresholding method. It only keep one lead SNP in each LD block, and we keep the marginal effect size as it is if like it is the significant lead SNP. And on the other hand, we have a more complicated method, the Bayesian method. So basically, it's kind of, you can understand it as a shrinkage method. It will keep multiple SNP in one LD block, and it will shrink the effect size of each SNP according to the LD information, and also assumption of what the true distribution of the effect size is. So this is the two, like the summary of these two methods, and I will just show one example of Bayesian method. Like mostly the Bayesian method just vary by they have different assumption about the true effect size. So the here is like I'll show the method that my supervisor Tiange developed. It's called PRS-CSX for continuous shrinkage, and oops, sorry, and like it adjusts the effect size, of course, according to the LD, and the assumption is that the effect size follow a normal distribution, and the standard deviation part contain two, like a group of parameter. One group is shows as the sigma squared is the overall variations, and the other is kind of like a shrinkage method that a shrinkage parameter that roughly you can understand this as it roughly represents the likelihood of a SNP being the caudal SNP, and that's like a parameter phi or psi. Sorry, I can't remember. So it follows a distribution called gamma gamma, and the shape of the gamma gamma you can see on the plot shows with this like a yellow curve, and compared with the normal distribution, we show it's in the dashed black line is have a sharper spike around 0 and a fatter tail on each side, so it can have a more realistic representative of like most of the SNP could be like just non-causal, but for the causal SNP, like the amount of the causal SNP may be like a lot larger than what the normal distribution would assume. Like if you're interested, like I can just let you know, like after you have this assumption, you may just like do the calculation using some like a mathematical like trick, like a MCMC, Monte Carlo Markov chain, or other methods. But I'm not going to spend more time like to explain all this like mathematical trick. So in summary, to calculate the PRS, we mainly adjust the marginal effect size we observed in the discovery GWAS for the LD structure. We can use the most naive method, clumping and p-value thresholding, or more complicated method like Bayesian methods. and also improving PRS. So this is like a large part of my both PhD and the postdoc. So I guess we are going to, we already seen this plot for like almost 100 of times. So we know that the current GWAS is like overrepresenting the European population. But to get accurate PRS, we are relying on the GWAS of matched population. So all the like the non-European population are like suffer from a less accurate PRS accuracy, and the field has been like constantly working on how to improve the accuracy for non-European populations based on whatever data we can have. And the reason that we have this like a low transferability of the PRS is like, of course, the LD is different, and also we may have some population-specific causal SNPs that are not that are almost impossible to measure in European population, and also different population have different environmental factors and like went through different like a natural selection and extra. But here we mainly focusing on the LD difference, and I'll explain why the LD difference can lower the PRS transferability across population and how we can solve this problem. So why when we use, like, for example, the European population to predict a non-European population, the PRS accuracy just dropped that much. And a major reason is the LD difference. For example, in the clumping and the p-value thresholding, as we mentioned before, we use a significant lead SNP as a proxy of the causal SNPs. However, like because of the LD structure, the lead SNP are not necessarily the causal SNP, and also because the LD difference in different populations, the relationship between the lead SNP and the causal SNP could be different, and the different population can have different causal SNPs. So besides that, like the effect size of the lead SNP can suffer from the Wiener's curse, which is like inflated because we choose the most significant SNP. So the lead SNP set we selected from one population may not as relevant with that trait in another population. So that's why we can see this like the drop of PRS accuracy or low PRS transferability. So how can we do better than this? So one solution is just like we use some more complicated PRS method, for example, the Bayesian method, because this method can include more SNP in the genome. So like it will be less sensitive to like the inconsistency of the lead SNP in different population. And also we adjust the effect size, so we partially solve the problem of the Wiener's curse. And of course, we need to combine more GWAS data. We can think of better way to do the combination, or just combine more data, or we can include other information. So all this, like the combination part, and also include the more information part, I will just show you how we did that in the field by some example. So first, the better combination method, I'll just shamelessly showing my own work PRS-CSX. And also, I'll also mention another method that I find very interesting, super learning method. And so the PRS-CSX is an extension of the previous method, PRS-CS. And what it's doing is like we are trying to more efficiently combine information across different populations. So what's about combining the information across population? So we think that the naive combination method usually using some oversimplified assumption. For example, when we use when we have a very strong assumption about the marginal effect size, which is like when we run the meta-analysis of two populations, we actually assume that despite all this difference, these two populations have the same marginal effect size. And then, like after we have this meta GWAS, we can just like adjust the effect size according to, for example, either the LD of population A or LD of population B or some other like LD panel we made, and then we choose we can choose the best SNP from all these like method and get the final results. Or we can just have no assumption, which we will call it the multi method. So the multi method is just like because we have no assumption, we just view each GWAS as independent. We just run the whole PRS process as if we only have the information of that one population. And from each population's GWAS, we get a set of PRS. And finally, we do the linear combination to combine these two PRS. So as a summary, like the meta method kind of consider, okay, the different population have should have some shared genetic architecture, but the way they model this shared genetic architecture is not efficiently, but and multi-population just never consider that. But on the other hand, the multi method considers the LD difference and the population-specific effect size when the meta method didn't consider. So none of them are like good enough. And also, according to other research, we know that we should consider the genetic shared genetic architecture because from the previous results, we know that the genetic correlation across population of the same traits is usually very high. And so a better assumption is that when we do this combination, we should assume that different populations share the same codal SNP and the effect size are like kind of largely correlated. So here is how we model this better assumption. So just a recap, in the PRS method, we have all this stuff, like we consider the LD structure and we assume the true effect size follow this distribution, and the distribution contains two type of like parameters, and some parameters like models the likelihood of the SNP being causal. So, but for the multiple population, and if we use the multi method, like all the parameters are population specific. So in the PRS-CSX, we keep everything else still being population specific, but this parameter that model the likelihood, or roughly model the likelihood of a SNP being causal is shared across population. So then we have this like a method we call coupled prior, which basically means the assume the true effect size distribution are kind of like correlated for the different population. And so are you?
Yeah, can I ask a quick question with hopefully not derailing you about using this word population? I was wondering if you could define how does an investigator figure out what to call a population? When do you want to make it more granular? And when can it be helpful versus hurtful to lump versus?
Yeah, that's a good question. So for like just for the context of like calculating the PRS for now, like I'm talking about I would just say literally by population is just like a group of individuals that their LD follow a certain pattern, or at least like, of course, this is a very, very oversimplified explanation for the population, but just for the sake of calculating PRS here, this is how I would use this term. I admit it's kind of very, very oversimplified. Okay. Is that okay? So, so from the using this coupled prior from the discovery GWAS from each population, we have a set of adjusted effect size, and based on that adjusted effect size, we calculate the PRS, and we finally linearly combine these two PRS as a final PRS. And here is some results. We tested this method and compared it with other existing method back then using simulation data. We can see that for both, we simulate a scenario when we have a larger European GWAS and a smaller African GWAS, and we can see that for both predicting European and predicting Africa, the green bars, which represent the PRS accuracy when we combine the two GWAS, is in general more accurate than using the single population GWAS, and especially the African targets benefits most from the combination of the GWAS, and we can see that from all the different combination methods, our method PRS-CSX performed the best. And we also repeated this test in biobank data. So here we just show the relative accuracy compared with only using the European GWAS, which here. Oops. And we can see that by adding more and more population, the PRS accuracy increase, and also we can see that our method PRS-CSX performs better than other methods. But also, as the author of this method, I know this method or this type of a combination method have its limitation, because when we combining more than one trait, or when we combine, say, 5 populations, the shared causal SNP assumption may not hold, because the more population you get, the more likely you will have some population-specific causal SNP, and of course, for different traits, the causal SNP should be different. And also the computation time will also increase when we include more GWAS. So if say we want to combine, say, 20 GWAS, the computation time of PRS-CSX could be quite long. So that's the limitation, and how to fix that I'll mention later. And also I will also mention some other method that can do the better combination, which is a super learn.
Can I ask a question before we move to super learning methods? This is online. Looking at the meta or multi, the different methods, it takes into account the different populations, but what about differences in the GWAS design, like the way the cases in control were selected? Does it account for those differences as well? And Sadi, please correct me if I asked that incorrectly.
The
Because it's not just the case, it's not just the populations that are different, the
Yeah.
selection of cases and controls or the imbalance, like the way the GWAS was designed might be different between those, and that might cause some heterogeneity between the two.
Yes, I totally agree. And for like, for example, like I'm not quite sure whether like this question is about how should we address that question. So I admit that that problem exists, but so maybe like like if you really concerned that the marginal effect size may really not be that consistent, maybe you should try more to the multi-method or the PRS-CSX method that can better model the like GWAS specific effect size. Is that what? Okay. Yeah. And if I'm not quite sure I can really address all this question very efficiently. So if you want clarification, please feel free to email me. Okay.
I have one more online.
Okay.
Are there any PRS methods right now that are based on statistical fine mapping, for example, from Susie, since those variants might are more likely to be truly causal?
I remember there's a method, but I didn't put it into my slide. There is a method like using the fine mapping information. Yeah.
Thank you.
And also the super learning method is kind of like just so to be brief, it's like conventionally we only choose the best set of adjusted effect size from each GWAS, but the super learning is like we can is combine the different set of adjusted effect size to further increase the information we can like extract from each GWAS. So that's the point, and this is one of the methods that use the super learning methods. And okay, I'm going to address what if we want to combine, say, like a few dozens of GWAS. So for now, like we in our lab, like we developed actually one type, but two different methods to do this combination. And I'll just show one example, like a GPS mode. So basically it's like do the combination in two layers. First layer is like we combine like say 5-ish different GWAS from different population, but of same trait together. And the second layer is like from each trait PRS, we then do the combination, and within each layer, we first do a feature selection, which you can understand that as like we remove some redundant underpowered GWAS from the model, and next we do a linear combination of the GWAS based on the selected powerful GWAS, and the result is that by doing this like all this combination of methods which show in the red dot and bar, like is more accurate than previously published PRS. And also we can try to include other information. So there are several. I'll just give two examples. So basically, by including other information, I mean we can treat the different region differently. For example, give some region a higher weight based on the information of like a gene set annotation, biological function, and so some actually we can be creative and thinking about all this information to be included. So the PRS per set method is like doing give the PRS weight slightly differently according to the gene set annotation. And also there's another method called X-wing, which includes the local genetic correlation into the model, which means like the more similar part of the genome across different population should treat differently than the less similar part of the genome. And so as a summary, so to increase the PRS accuracy for diverse populations, we combine information from different population, different traits, annotation, and all these other informations. And also the final part, the standardizing and applying the PRS, which we have some good news and bad news. The good news is like a PRS can really do a lot of things. So I listed several applications of PRS in like a basic scientific research. We can use the PRS either based on the whole genome or based on part of the genome to predict the disease or subtype of the disease. We can use the PRS, so we can use the PRS as instead of the SNP as a proxy of to be the instrument variation in Mendelian randomization, and we can use the PRS to analyze all this interaction either with the environmental or like the rare variants, and there could be even more as the field developed. And also how close we are to apply the PRS in clinic. There are some also very promising results showing that we can really use the PRS to help in the clinical trial to see that the PRS can help us to identify the younger individual which around the borderline or in the intermediate clinical risk to determine whether they should start therapy earlier than as suggested by conventional prediction model. And also we have some kind of like a bad news. It's like the current PRS is kind of like cohort specific. If we're just looking at the raw PRS value, it's kind of like it only makes sense in each specific cohort. For example, if we calculating the raw PRS across different population, we can see that even like our everyday experience telling us maybe like the African population and the European population roughly have similar age height, but the PRS of African populations can be much lower than the European population. And also our data tell us like across the whole globally the prevalence of the schizophrenia are quite consistent, but also the distribution of raw PRS could be like very, very wildly. So we cannot directly use the raw PRS across cohort, which can be a problem. And also the PRS weights have inconsistent accuracy in different populations, as we mentioned before, the low transferability. And here is another represent is that we test the same PRS weight, and we can see the accuracy just drop almost linearly as the genetic distance increase. And there are some way we can try to maybe partially fix the problem. So for example, the different population have different raw PRS distribution. We can just regress all the PC so they have more uniform distribution. And lately, I developed a method that can that interpolate PRS across the whole spectrum of the PCA. So instead of like just like fine-tuning the PRS model in the matched population, I fine-tune the PRS in different single ancestry cohort and use their PCA information to interpolate the PRS for diverse admixed individual. And in our test, we can see that, so this plot shows the relative increase of the interpolated data versus the PRS created by the conventional data. We can see that the interpolated data have similar and sometimes slightly better accuracy than the PRS like created by the conventional data, which is like fine-tuned with the matched population. And if you're interested, here is the preprint. So yes, we can partially address this inconsistency, but still like they are so this is like just addressing the inconsistency using the PCA information. However, like some other research shows that the PRS performance can be influenced by other factors. I will just show here the kind of take-home message from this paper. Sorry, part of the take-home message. So the context such as like age, sex, income can impact the PRS accuracy with the same magnitude as the genetic ancestry. So there are all these factors we need to consider if we want to further increase the PRS prediction accuracy. And also, there are some other limitations. So despite that we have already some like almost saturated PRS for the quantitative trait like height, but for a lot of complex disease, the PRS accuracy is still quite limited. And also, we also notice that the PRS can be very unstable on the individual level. So here I list two papers that talking about this phenomenon, which means like even on the population or cohort level, like maybe they have very similar R squared, but the high risk individual they predict can be varied very differently. So we still need to address this problem if we want to have better or more stable PRS, which is more suitable for clinical usage. And okay, here is the summary of all the talk. So the last part is like, yes, good news is that PRS have all this potential for the predict within the cohort and have many useful application in both basic scientific research and probably very soonish clinical usage. But however, like more works are definitely needed to improve its stability and widen its application. And I will thank all my wonderful supervisors and all my co-workers and friends that give me advice and discussing all this problem with me. And thank you for listening. Any questions?
In the meantime, I was wondering if you have suggestions for sample sizes or heritability explained to have a reliable PRS, and if there are dangers or things to look out for when calculating these in smaller or underpowered GWAS.
Like what is the, do you mean what is the smallest sample size for creating?
Yeah, like if I had, if hypothetically I studied a rare disease and I don't have like the heritability explained and the numbers like a height GWAS would have. do you feel like there's recommendations of the heritability explained or the number of people you would want in your GWAS to have the PRS be more reliable?
That's a good question. Like for like I think that depends on like your aim for the analysis. It's like if you want to just show whether you can, if you want to just see the whether there's a connection between the PRS and the outcome traits, maybe you can expect like a quite small GWAS made already enough to show some like a correlation, like in this example. But if you want to say have a more stable, nice result, maybe you need a larger sample size. And one thing, actually, like the actual number is quite difficult to say because it have to like how the prevalence, the genetic architecture, basically like we would say the more the better. And another thing is like if you really have very small sample size of the matched traits, you can use correlated traits to kind of like increase the power. Like for example, in our GPS mode method, when predicting the CD, we also use like say blood lipid BMI to increase the power. So that's some way you can use to increase the sample size, because basically my answer will be the more the better.
Hi, thank you for a great talk. I'm wondering, this context specificity that like Bogdan's recent paper shows seems like really hard to know how the field's going to get around that. And I'm wondering what your views are for making progress on that front. And by that, I mean like the fact that the prediction accuracy just varies so much by all of these non-genetic factors.
It's like so my answer may be not the best answer, but that's how I see it. So if, of course, like there are going to be the interaction between the GWAS, between the genetics and the environmental, and I think it's kind of like it just sucks because we want to just use as simple prediction model as possible, because like we can see all this like the PRS with the performance would vary so differently and within all this cohort. But I think it's kind of inevitable because I think that's how complicated the G by E interaction should be. Maybe like for my current understanding is like maybe we just try to cover the genetic part as good as possible, and viewing all this like complexity of G by E caused the problem as an opportunity to study the G by E. That's my current understanding.
Right, and then does that maybe suggest that as a field we need to stop thinking so much about predictive accuracy, stop gobbling up the environmental part of the variance?
Like not exactly. I think at least my understanding of the instability, I think is still caused by the GWAS is underpowered when predicting all this like SNP level statistics. So, and that's why that's probably like one of the main reasons why we see when we're using different GWAS method, like the adjusted effect size could be that difference because that's comes from the method and also the low power of SNP level stability. So I think maybe the instability can be partially solved when we have more powerful GWAS, and then like that can, at least we can fix one thing. We can like have one thing fixed and to see better pattern of how all these factors interact. That's my understanding.
Great. Also just wanted to say I love the name Disco Divas for your method.
Thank you.
Amazing. We're at time. So thanks everybody. Again, reminder that next week is meeting free week, so there's no meeting, and we'll see you in 2 weeks. Stay warm. Thank you.